One thing that really needs to be dealt with is vaccine misinformation. I'm sure we can all agree on that. It's crazy to me still how there is still so much misinformation online, and there are still approximately 30% of adults that have yet to be vaccinated. If you're in the US, I'm sure you personally know a friend or family member who is subscribed to misinformation. There's this video that was sent to me last week, and it has over 2 million views. I don't understand. Why is it that crazy videos like this get so many views? It almost feels like these conspiracy theorists dominate social media or something. I don't know.
Hello everyone, this is Dr. Mikola Rashik of Mura Genomics, once again making an update video on the mRNA vaccines. And today I probably have one of the biggest and wildest news I can share with you when it comes to molecular biology impacted by either COVID-19 or vaccines.
By the way, in case you haven't read the news lately, anyone in the US now can get a third shot of the vaccine. I don't know about you guys, but I'm going to be rushing to the nearest pharmacy. I can't wait to get my third dose of microchip. That way, if I get lost in the middle of a forest, Bill Gates can come save me.
Definitely a game changer, so let's get started. Recently, just about a few weeks ago, this was in the first week of October, a research team from Sweden published information that showed in their human cells that they were studying that the spike protein can actually enter nucleus.
See, I'm doubtful of something like this because proteins that bind to the surfaces of your cells usually can't pass the plasma membrane. Especially if this protein is attached to a virus, I don't see any reason or any mechanism in which it would enter the nucleus. But I guess it's technically possible that after the virus enters the cell, when it's manufacturing new viruses, that the spike protein inside could travel into the nucleus. Of course, I had to find this study myself, and it seemed to have come from this paper titled SARS-CoV-2 Spike Impairs DNA Damage Repair and Inhibits VDJ Recombination in Vitro. Let's start off by reading the abstract. Severe acute respiratory syndrome coronavirus 2 has led to the coronavirus disease 2019 pandemic, severely affecting public health and the global economy. Adaptive immunity plays a crucial role in fighting against SARS-CoV-2 infection and directly influences the clinical outcomes of patients. Clinical studies have indicated that patients with severe COVID-19 exhibit delayed and weak adaptive immune responses. However, the mechanism by which SARS-CoV-2 impedes adaptive immunity remains unclear. Here, by using an in vitro cell line, we report that SARS-CoV-2 spike protein significantly inhibits DNA damage repair, which is required for effective VDJ recombination in adaptive immunity. Mechanistically, we found that the spike protein localizes in the nucleus and inhibits DNA damage repair by impeding key DNA repair protein BRCA1 and 53BP1 recruitment to the damaged site. Our findings reveal a potential molecular mechanism by which the spike protein might impede adaptive immunity and underscore the potential side effects of full-length spike-based vaccines. So basically, the authors claim they find a mechanism in which the spike protein localizes in the nucleus and inhibits core mechanism of adaptive immunity, which by the way involves DNA repair and VDJ recombination. We're going to critique this in more detail in just a second, but I see their motivation in this study, which is that patients suffering the most from COVID seem to have weaker immune responses, so they want to find a mechanism in how this is happening. Very reasonable. Okay, so the rest of the video is this guy drawing his own conclusions based off the results of the study. So before I get to his points directly, I'm going to talk about this paper, which is the source of all his claims. I've seen a few times on the internet regarding people referring to this Swedish paper, saying, look, this paper proves that vaccines impairs your immune response. The government is trying to use us as guinea pigs. Just another piece of misinformation to promote vaccine hesitancy. Okay, so I went through and read the paper so you don't have to, because we all know how boring scientific papers are. The authors studied this mechanism in HEK cells in vitro and grown in a culture, treated with DNA damage reagents with controls of course, and observed under fluorescence where it was ultimately analyzed using t-test and ANOVA. Spike protein was allowed to be produced in the cytoplasm, and the authors observed a significant amount of it in the nucleus, where it inhibited DNA repair and VDJ recombination. I won't actually go into any more detail regarding the methods used and how they observed the results, but if you're interested in the biological procedures, the paper will be linked in the description below. Anyway, the discussion has a few comments about the current vaccine, saying, this suggests that the use of antigenic epitopes of the spike as a SARS-CoV-2 vaccine might be safer and more efficacious than the full-length spike. And this is where all the conspiracy theorists jump in, because they read news articles that interpret this paper and jump on the bandwagon against vaccinations when they haven't actually read the original paper. And we also get videos like this one we're responding to, propagating this kind of misinformation. Don't get me wrong, this isn't a case of news media outlets misinterpreting the paper. Oh no, this is a case of the paper itself being dubious. Now, I have no problem with all the specific techniques they use, but the authors really had some questionable methods in their experimental molecule's design. There are two major points I want to make. First of all, the authors used HEK293 cells, which in case you guys don't know are kidney cells. Now, there has been evidence that COVID-19 affects the kidney, but you have to remember we're talking about the adaptive immune system here because our area of focus is on the spike protein's effects on antibody production. In other words, the choice of using kidney cells is crazy. For some reason, the researchers decided not to use relevant cells. Even using epithelial cells in the lungs is not the best because although it's the primary target of the virus, they're not responsible for immune responses. This brings me to my second point, that the paper fails to understand the activation mechanisms of the adaptive immune response. Remember, B cells don't get activated from being exposed to the virus itself. Instead, you have antigen-presenting cells which engulfs the virus first, then breaks down its antigens and presents it to the helper T cells. Keyword being break down because you won't have a full-length spike protein at this point. The helper T cell then travels through the lymph node to reach the multipotent progenitor B cells. It then finds the B cell that matches the antigen in question, and that B cell is able to proliferate. So in other words, the progenitor B cells itself here will never really be exposed to a virus or a full-length spike protein. And even after they proliferate and enter the battlefield, by then their antibodies have already been developed, so there's no point infecting them after proliferation to impair immune response. Meanwhile, the vaccine uses a similar but slightly different mechanism where instead of an antigen-presenting cell engulfing a virus, the RNA directly enters cells and skips that first step entirely, but still presents antigens to the helper T cells. By the way, that's why getting an RNA vaccine does not leave free-floating spike proteins in your blood. I can go into the adaptive immune system pathway in a bit more detail some other time, but the main takeaway here is that even if the full-length spike protein does have such an effect on DNA repair and VGJ recombination, the paper's methods show a lack of understanding of the primary pathway in which the adaptive immune response is activated. In addition, they also chose kidney cells for some reason, so that's very strange already. A bigger takeaway message here is that not all scientific papers are of good quality. Make sure you actually read the paper itself, investigate its methods, see if it's peer-reviewed, and compare it to already sound scientific knowledge before blindly accepting it as true. Which is why I love scientific organizations such as the CDC or WHO, because sometimes I just don't have time myself to comb through all the recent scientific research on COVID-19, but they have scientists that specifically do this. That's why I knew before even reading the paper that it's nonsense, because otherwise at least one major public healthcare organization would have commented on it already. If the RNA vaccines really did impair immune response, the vaccine would have been halted immediately. But yet we have paper after paper being released showing the efficacy of the vaccines. This paper is not an excuse to not get the vaccine. Get your shots or I will inject you in your sleep. And of course, stick around for 15 minutes in case you get an allergic reaction.
And the reason why this is such a dramatic news and why it's such a game changer is the fact that obviously nucleus houses our genetic material, all of our DNA, almost all of our DNA. Mitochondria is the other place where you might find DNA. And what they also showed, and normally entry into the nucleus is supposed to be verboten, you're not supposed to be going inside there, precisely because you could potentially be modulating how we're using our genetics. Now, what did they discover is that once the spike protein enters the nucleus, it actually inhibits proper fixing of broken, damaged DNA, and the specifically damage of DNA where you introduce double-stranded break, meaning really you break both strands of the DNA, you shear the DNA in half, you break it in half. And the way it does negatively impact how the DNA is being fixed is by somehow, it's still unknown, somehow interfering with how BRCA1 protein is supposed to be used in fixing that DNA damage.
Well, actually, the paper does tell us. It impedes BRCA1 and 53BP1 recruitment to the damaged site. Anyway, you can kind of see where this video is going, and I'm going to cut bits and pieces out of it because he takes a long time to make certain points, and some of it involves him explaining the functions of specific proteins involved in DNA repair, which we don't actually need to know for the purposes of this video. I can tell this guy is indeed an expert in his field, which is why I'm confused why he made this crazy video.
So why do the authors suspect this might be happening? They believe that potentially this is how the virus evolved this method in order to inhibit how our immune system responds in being able to combat infection. So keep in mind that when we produce antibodies or T-cell receptors, they can recognize pretty much any pathogen. So how do you achieve that? Well, the cells that are actually responsible for producing either the antibodies or the T-cell receptors, they can actually shuffle the genetic material that is responsible for production of these proteins, the antibodies or the T-cell receptors. So it cuts the DNA apart and then splice it in a variety of different manners, and that's how you can literally produce any amount of these proteins being able to attack any pathogen.
Yeah, so this is actually why your adaptive immune response is literally able to fight anything and everything. It does so through a recombination method that allows it to produce a number of combinations of DNA, which then makes the antibodies required to fight pathogens. Interestingly enough, I actually made a video on this exact topic a long time ago, over 2 years ago, but you guys never actually got to see it. For those of you who have watched me for a long time, you probably remember when I announced a second channel, but it never actually came out. There, I was going to make a show similar to SciShow, where I explained various scientific concepts. But due to the workload and my team being lazy, you know who you are, the first video was never released and thus the channel never took off. That video was actually going to be on this VGA recombination topic. Ah, such a shame really. Okay, back to reality.
So by spike protein entering the nuclei and preventing this from happening, what it actually in essence would be doing is preventing the development of diversity of response to the infection. So why is that potentially dangerous? Obviously, is that in theory, then the spike protein could be a mutagen.
Viruses causing cancer is really not a new thing. They're called tumor viruses. The vast majority of cervical cancer is caused by HPV. It makes sense because viruses would want your infected cell to proliferate more. That way the virus itself could proliferate more. But even so, it's not always advantageous to do so for reasons I won't explain in this video, which is why the majority of viruses aren't tumorigenic. Okay, we've delayed this long enough. Let's go to the part where he talks about what this means for vaccines.
It can take many years before clinical symptoms show up, and that actually brings me to another point, is the whole concept of mentioning that vaccines are safe. The actually more proper way of saying is the fact that we actually do not know what the vaccine spike proteins will do in our body post-vaccination, but on a short-term scale, they do not frequently produce clinical symptoms. That's the more real explanation. Not that they're safe, it's just that we don't know what they do, but they do not produce dangerous clinical symptoms in the first few months.
Okay, so to summarize, he's drawing his own conclusions from the paper and decides to focus specifically on the part of the paper's findings on DNA damage repair. He doesn't focus as much as what this means for the immune system, but rather hinting that these mechanisms could cause cancer for your cells. Then, since the vaccines use full-length spike protein, it could potentially have long-term effects regarding cancer development. Now, I'm not going to entirely dispute this claim regarding what the spike protein could do in terms of mutations. However, he's trying to link this to vaccines instead of the actual infection. The authors of the paper here used kidney cells, so if the findings are correct, then the actual virus can cause DNA damage. However, the vaccine localizes in muscles near the respiratory system, which is essentially different. Of course, that doesn't necessarily throw away that particular finding of the paper. A lot of cells can be similar in many ways, but it does undermine its link to vaccines. If anything, you proved more so that the virus can cause this type of damage, not the vaccine. That's a reason to get your shots, actually. In addition, we know that RNA and the spike protein by itself does not last long in the body, not more than a few days, because your body breaks it down rather quickly. So even if all your hypotheses are true, there's no good reason to believe that vaccines can cause long-term damage to the body. The virus, on the other hand, very well can, because the spike protein attached to a virus can cause a ton of damage and last a very long time in your body. It really seems like the scientists took everything into consideration when creating the vaccine, didn't it?
Now, another thing that I should mention is the fact that these authors mentioned that it requires a full-length protein, full-length spike, and that's exactly meaning entire protein. It's not a fragment of the protein, it's entire protein enters nuclei and causes this, and it facilitates or helps this damage to the DNA. Now, remember that the vaccines are indeed using full-length protein.
I'm not against using a particular epitope, such as one specifically to the receptor binding domain, but I also don't see a good enough reason to do so. If the RBD mutates to produce a new viral strain, your vaccine would lose efficacy faster than my wallet loses money on the upcoming Black Friday sales, and I am known to trample over children to get my $2 off on a shiny new can opener. Speaking of which, make sure you all stay safe during the holidays, and if you see any Christmas decorations before the end of Thanksgiving, be sure to throw a tantrum. That's all for me. Thank you for watching, and thank you to Fireshard, Alan Morton, and Miss Fixit. See you later.